In video 4 of this unit, we're going to be exploring mitosis and how it's related to tumors and cancer. So therefore, today's video will be focusing on our last focus question, which is what is the relationship between mitosis and tumors? In yesterday's video, we saw in the cell cycle there are many checkpoints where we check to see if the cell is ready to move on to the next stage. So if cells become unresponsive to these signals and they move on to the next stage of the cell cycle when they're not ready, that's when we have cancer. So in cancer, we're basically getting excess division. We're getting cells dividing when they're not supposed to. So we end up with a bunch of cells kind of piled on top of one another that have divided too much. And this is called a tumor, when we have a bunch of cells, a clump of cells that have divided too much. Oftentimes, we get so many cells built up in this tumor that those cells start to leave and invade other tissues, and this invasion of other tissues is called metastasis. And finally, the combination of these two things, so cells dividing when they're not supposed to, excess division, and metastasis, often lead to death of the organism. So remember from yesterday that a normal cell, or a non-cancerous cell, only grows when it's on a solid surface, so we say it's anchorage dependent. And it also stops growing when it collides with other cells, and we call this density dependent inhibition or contact inhibition. So cancer cells are a little different. Because they're now ignoring the signals, the checkpoints in the cell cycle, they no longer exhibit anchorage dependence, so they do not need to be on a solid surface to be dividing, and they no longer exhibit density-dependent inhibition. So now cancer cells will divide even after they've come in contact with another cell, which is why we get the formation of a tumor. So you may have heard before, there are two types of tumors. So the first is benign tumors, and these are tumors that are considered to be non-cancerous, and benign means not harmful. So these are the tumors where perhaps you're getting too much growth or too many rounds of cell division, but the cells themselves are not that harmful. They don't have that many mutations, so they look and act like normal cells. These cells also cannot infinitely divide, so they're dividing more than they're supposed to, but they're not going to divide forever. And finally, they're also going to stay in one place. They're not going to metastasize or travel to other parts of the body. On the other hand, a malignant tumor, this is what you usually think of when you think of cancer. These are cells that are not only dividing way more than they're supposed to, but they also have other mutations that make it so they cannot do their job. So these cells, because they have so many mutations, they're often the wrong shape. They have mutations that make them lose control, and they're doing way too much division. They're dividing when they're not supposed to. And they also have mutations that allow them to travel to other parts of the body and invade other tissues. So in order for a cell to be malignant, or a tumor to be malignant, two things have to be true. So first, the cells have to be mutated to the point they can't carry out their normal functions. So they have some mutations that make it so they cannot do their job. And these cells have to be mutated to the point that they're ignoring the signals in the cell cycle. So they're dividing too much. And because they're dividing too much, they crowd out normal cells, so they take up the space that normal cells should be taking up. And they also use up the nutrients that normal cells need. So because there are so many of them, they're taking up vital nutrients, and the normal functioning cells cannot get the nutrients they need. We've seen so far in this video that cancers are caused by mutations in the DNA. And things that cause mutations that result in cancers are called carcinogens. So some carcinogens you might be familiar with are so UV radiation. So if you're out in the sun too much, the UV light in sunlight can be very damaging to DNA and can cause mutations that can lead to cancer. Similarly, chemicals in tobacco and other toxins and chemicals can interact with DNA and cause mutations. And even some viruses, like the human papillomavirus or HPV, can interact with DNA and cause mutations. So in your DNA, there are two types of genes that control the cell cycle. There are proto-oncogenes and tumor suppressor genes. And these are the genes that create those cyclin proteins that are responsible for the checkpoints in the cell cycle. And if you have a mutation in one of these genes, your cells are going to divide uncontrollably because you no longer have a functioning cyclin to control these checkpoints. So the first type is proto-oncogenes. And these are genes that promote growth. So these were those proteins in the cell cycle that if everything was okay at the checkpoint, they said, yep, you're okay, go on to the next step of the cell cycle. So normally, in a normal situation, I can turn these genes on or off. If my cell is not ready to go to the next step of the cell cycle, this gene is off, and I'm not producing the protein that would allow it to go to the next step. If the cell is ready to go to the next step of the cell cycle, this gene gets turned on, and it produces the protein that allows the cell cycle to continue. However, if you're exposed to too much UV light or too much tobacco or other carcinogens, and a mutation is introduced into one of these proto-oncogenes, the cell gets stuck. So now cell division is always on. This gene gets stuck on. And when this happens, this gene is no longer a proto-oncogene, but it's now what's known as an oncogene. So an oncogene is the mutated version of a proto-oncogene. So if we have a mutation that turns these genes always on, now we're always going to get cell division, even if we're not ready for it. The cell keeps dividing and dividing and dividing, whether it's ready or not. The other type of gene that if we get a mutation will cause cancer are tumor suppressor genes. So these are the genes in the cell cycle. If the cell is not ready to go to the next stage, these genes produce proteins that say, no, stop. Something's wrong, you can't continue. And if we have a mutation in these genes, basically cell division is never stopped. So even if there's an issue in the cell, it continues to divide and it continues to grow. So you can see how if we have a mutation here, we get excessive growth and division in cells that were not ready to do so. And this would also cause cancer. So an important thing to note about cancer is it takes more than one mutation usually to cause a cancerous cell. You first need the mutation that allows for excessive cell division. So you first either need a mutation that would, in either a proto-oncogene or a tumor suppressor gene, that would allow the cell to divide uncontrollably. Then you need a second, third, fourth mutation that will cause the cell to no longer be able to perform its function. So if we look at this picture here, maybe my first mutation, the one listed right here, was in a proto-oncogene. So now my cell is always dividing. So I'm getting basically a healthy cell that's dividing way more than it's supposed to. And then maybe later in its lifespan, I get a second mutation and this mutation doesn't let it do its job properly. So now I have a cell that's dividing too much that can't do its job. And then I get another mutation that makes my cell even more useless, so it can't do a second job. And maybe a fourth mutation. So now I have a cell that has many issues that keeps dividing and is taking up space and nutrients from healthy functioning cells. As you might be aware, there are many different types of cancers, and an important thing to note is that cancers are named for the organ or tissue in which they originate. So if a cancer is called a carcinoma, that means the original mutated cancerous cell originated in either an external or internal covering of the body. So maybe it originated in the skin, for example. In a sarcoma, the original cancerous cell arose from a support tissue. So either maybe you had a mutated bone cell or a muscle cell. And in either a leukemia or a lymphoma, your original mutated cell came from a blood cell. So a bone marrow cell perhaps, a cell in the spleen, or in the lymph nodes. 5 things that all cancers have in common. So whether it's a sarcoma, a leukemia, all cancers show these 5 things in common. So one, most importantly, the biggest hallmark is uncontrollable cell growth. We're getting cells to grow when they're not supposed to. Usually we also see what's called death evasion. So every cell, when it first comes into existence, has a programmed amount of time that it's going to exist before it's going to be recycled. Cells over their lifetime usually gain mutations, so we only want cells to be around for a certain amount of time before we get rid of them. Cancer cells have usually figured out a way around this, so even when they're supposed to be recycled because they're too old, they usually continue to divide and live on. So this we call death evasion. We also see immortalization, this fourth point down here. This is kind of similar. Basically, cancer cells can divide an infinite amount of times. Usually, cells can only divide a certain amount of times before they're recycled, but cancer cells can kind of divide infinitely. We also see angiogenesis, which is basically when cells attract blood cells to themselves so they can get more nutrients from the blood and get rid of waste more easily. And this is a problem because basically they're stealing the nutrients from the healthy cells around them. And then finally, we also see what's called metastasis, which we've already discussed. And this is the ability of cancer cells to spread to other parts of the body and form new tumors there. So this last picture is a picture of metastasis. So you see I have a benign tumor, so a cell that doesn't have too many mutations but is growing out of control. And eventually, what can happen if you get too many cells to build up, it can break through the layer that separates these cells from a blood vessel. And if these cells enter the blood vessel, they can then travel to other parts of the body. So in this case, we're moving from the skin to the liver. And they can kind of establish a new tumor there and grow as well, and maybe obtain more mutations and become more malignant.